This section explains why the whole first chapter turns on a single inversion. Shrink the instrument onto the specimen instead of pinning the specimen under the instrument. That inversion is the entire chapter. Benchtop microscopy demands an anesthetized, stereotaxically restrained brain, which makes behavior a confound rather than a signal. Anesthesia itself distorts excitability, blood flow, and connectivity. A head-mounted microscope light enough to ride the cranium dissolves that constraint. The animal explores, mates, runs its maze while its neurons are watched, so activity and behavior become co-observable. As the handbook's opening chapter, it names this as the design axis. Every later modality, single photon, two photon, speckle, photoacoustic, is the same instrument onto animal pattern carrying a different contrast. One boundary remains, the tether. Power and acquisition still live off-head, so freely moving is not yet untethered. This section explains how the physical mounting of an imaging instrument sets the ceiling on what an experiment can ask. Shrink the microscope until it rides on the skull and the animal's restraint dissolves with it. The way the instrument mounts fixes the experiment's limit. A benchtop system demands an anesthetized, head-fixed animal. Anesthesia flattens the very dynamics you wanted to see, and restraint severs neural activity from behavior. Miniaturize the optics into a headpiece of about 2 grams, and the freed variable, natural behavior, mating, exploring, locomoting, becomes the newly imageable signal. Correlated frame by frame becomes the newly imageable signal. Correlated frame demands. The chapter opens the handbook on this move. What the platform physically permits the subject to do bounds what neuroscience can ask. The residual constraint is the tether. Even discrete systems still trail a wire to power and storage. Truly tetherless imaging stays the open boundary. This section explains the single idea behind functional imaging with light sheet microscopy. The whole chapter is one move. Illuminate only the plane you are already watching. The excitation beam runs perpendicular to the detection axis, so the light arrives as a thin sheet rather than a point or a full cone. That one geometric choice is everything. Contrast this with confocal and point scanning methods. They flood the entire depth of the sample just to read back 1 voxel at a time. Light sheet excites exactly the section. The camera images and nothing else. The lit region and the image region become the same region. From that single decoupling, both prizes fall out together. The camera reads the whole lit plane in parallel, which buys speed orders of magnitude faster than point scanning. And no fluorophore outside the imaged plane is ever bleached or poisoned, which buys gentleness, hours of live neural recording. Volumes come from sweeping the sheet through depth. That is the chapter's argument. This geometric decoupling is why light sheet, rather than confocal, can image a firing brain whole. This section explains the core idea behind light. Sheet microscopy. The principle is one sentence. Illuminate only what you are currently reading, that is, the single plane you care about. The whole chapter. Conventional fluorescence. Microscopy, whether confocal or epi, floods the entire depth of the specimen just to recover a single in-focus plane. Every photon spent outside that plane is pure cost. It buys you photobleaching, phototoxicity, and scattered background, and nothing else. Light sheet rotates the illumination so that it sits orthogonal to detection. A thin sheet then lights exactly the plane. A wide field camera captures and you build volumes by sweeping the sheet through the specimen. That single geometric decoupling is generative. The speed gain of orders of magnitude, the gentleness that lets you image a whole zebrafish brain live for hours, the depth penetration, all of it falls out of that one choice. The 5 performance parameters all compete for one shared photon budget. The sheet's job is to waste none of it. This section explains why a 2-photon microscope can see deep into living brain tissue, and it traces every feature of the instrument back to a single physical fact. The excitation scales with the square of the light intensity, so it fires only where the photons pile densest at the focal spot. That square law dependence buys the microscope its optical's point, where the beam converges to its tightest stray light. With no pinhole to reject stray light, one dye molecule must wait for 2 half-energy near-infrared photons to arrive within about half a femtosecond of each other. That coincidence is vanishingly rare everywhere except at the focus, where the beam converges to its tightest point. The whole chapter descends from this one primitive. Because only the focus is bright, you must visit points serially, one at a time. And because the light is near infrared, the photons ride the tissue's optical window deep into the brain before they scatter. Every downstream engineering problem follows from that same gate. The slow, voxel by voxel scanning and the out-of-focus background that grows with depth are both the price you pay for the square law rule that made the sectioning possible in the first place. This part explains how squaring an excitation confines it to a single point, and what that confinement costs. Squaring an excitation confines it. That is the whole move. Make fluorescence depend on the square of light intensity, and emission collapses to where intensity peaks, the diffraction-limited focal spot. Everywhere else, the intensity is too low for 2 photons to arrive within the roughly 0.5 femtosecond coincidence window. A global floodlight becomes a 0.5 femtosecond coincidence. Depth follows for free. Near-infrared sits in tissue's optical window, and since only ballistic photons at the focus make signal, all light can be collected with no pinhole, pushing imaging from roughly 100 micrometers to a millimeter deep. The bill comes due as time. 1 lit voxel means the volume must be scanned serially, slowly. The chapter is then the engineering of buying speed back against that constraint. This section explains why light field microscopy can capture a whole moving brain in a single snapshot. Start with what an ordinary microscope discards. It focuses rays by their point of origin, and in doing so, it averages over the directions those rays were traveling. That averaging over direction is exactly what blur is. A conventional microscope throws away where each ray was going. Light field microscopy keeps the direction. Place a microlens array at the image plane, and each sensor patch records not only where a ray landed, but the angle at which it arrived. The full 4-dimensional light field, the function L of SST, U, and V, is captured in a single exposure. Depth stops being something you scan for and becomes something you compute. 1 snapshot fixes the whole volume at once. This is why acquisition scales to whole brain rates, around 100 Hz, independent of the size of the volume. The price is fixed. Angular information is bought with spatial pixels. This section explains how light field microscopy captures depth from a single snapshot. Blur is discarded direction. A camera focuses every ray from a point back onto 1 pixel, averaging away the angle each ray carried. An angle is precisely what encodes depth. Light field microscopy keeps the angle. A microlens array at the image plane splits each point into the subapertures its rays passed through, so the sensor records the full four-dimensional light field, written L of S, T, U, and V, position and direction together, where an ordinary camera retains only a flat, two-dimensional slice from a single snapshot. You then computationally refocus any plane, reconstructing a whole volume with no scanning. The trade is fixed, lateral resolution spent to buy angular information. The primitive, capture the entire ray manifold once. Choose the projection afterward. This section explains why activity indicators get written into the genome instead of delivered as dyes. Optical dyes report neuronal activity everywhere they diffuse. The readout is cell-blind, staining whatever it touches. The chapter's move is to stop delivering the sensor and instead encode it in the cell's own genome, so the promoter driving its expression doubles as a selector on which cells you listen to. Sensing and addressing collapse into one construct. Two things get reported. The first is calcium, bright and easy, but a surrogate. It is blind to hyperpolarization and to sub-threshold events because it only spikes with action potentials. The second is voltage, direct but dim. The chapter's spine is that trade-off. Genetic specificity is the prize, and every subsequent choice, GECI versus GEVI, each spectral and kinetic variant is negotiating what fidelity you sacrifice to keep it. This section explains how to read neural activity as light, and the one move that defines it, shifting the reporter from something you paint on to something the cell transcribes. That is the whole chapter. An applied dye stains every membrane it touches, neuron, glia, and vessel alike, so a population signal cannot say which cell class fired, and the dye is transient and toxic. Which cell class fired? Encode the indicator as a gene instead, and cell type specificity comes for free. The reporter expresses only where its promoter fires, stably, for months. Everything downstream is a choice of what to sense. Calcium is an indirect surrogate with a large, slow signal. Voltage is the direct readout that faithfully tracks fast dynamics at the cost of a smaller one. Specificity comes by construction, not by staining. This section explains functional optoacoustic imaging and why it reaches deeper than light alone can. Light dies in tissue, not because it stops, but because it scatters. Past a millimeter, the photons diffuse and resolution collapses. Optoacoustics escapes by changing the readout channel. Pulsed light is absorbed. It heats a chromophore. The flash expansion launches an ultrasound wave, and you record that wave. Sound scatters 1,000-fold less than light, so you keep optical absorption, contrast, hemoglobin, oxygenation, melanin, injected reporters, while inheriting ultrasound's reach centimeters deep. The chapter names it superhearing. The generative move is the transduction itself. When a medium scrambles your signal, convert it into one. The medium leaves alone. Same information, quieter carrier. The residual limits, the skull, light attenuation, are exactly where the carrier also gets scrambled. This part explains optoacoustics as a structural primitive. Light goes in, sound comes out, and the whole chapter turns on one refusal, not reading the signal back through the channel that degraded it. Optical microscopy dies at roughly 1 millimeter because photons scatter and resolution dissolves into diffusion. But the absorption contrast that light creates, in hemoglobin, in oxygenation, in melanin, is what you actually want. Optoacoustics keeps the optical excitation and swaps the readout to ultrasound, which scatters some 3 orders of magnitude less, so resolution survives centimeters deep. Rosansky's word for this is superhearing. Here is the generative primitive. When one channel's physics caps you, encode into it, but decode through a channel that does not share the limit, a hybrid that inherits contrast from one side and penetration from the other. This section explains how imaging deep in the brain turns, scattering from an obstacle into the instrument itself. Scattering in tissue looks like loss, but it destroys no information. Coherent light, passing through a fixed, disordered medium, undergoes a deterministic linear transformation, a transmission matrix that randomizes the field while keeping all of its information intact. Confocal and two-photon microscopy treat scattered photons as garbage and keep only the unscattered ballistic ones. So they starve exponentially and stall at the transport mean free path wall near 1 millimeter. Wavefront engineering makes the opposite move. It measures or probes the medium's operator with a deep guide star, then pre-shapes the incident wavefront to its inverse, so the scattering itself reassembles the light into a focus. The generative primitive is this: invert the channel, do not filter it. That is precisely adaptive optics correcting atmospheric turbulence. This part explains why scattering tissue can be beaten rather than merely endured. Scattering tissue does not destroy light. It is a fixed linear operator that scrambles it, and a fixed operator inverts. Every microscope before this chapter fights scattering by discarding the scrambled photons and keeping the ballistic few. That strategy decays exponentially, and it walls out near 1 millimeter. Wavefront engineering makes the opposite bet. You pre-distort the incoming beam so that the tissue's own scrambling cancels at the target, collapsing multiply, scattered light back into a focus. A guide star deep in the brain supplies the feedback that fixes which pre-distortion to use. The whole chapter is one move. You turn the enemy, transform into something you compose your input against, exactly as astronomers unshape starlight through the atmosphere. This section explains how imaging below the resolution limit of light turns synaptic biology into a geometry. When you make the microscope see below light's own limit, synaptic biology becomes a geometry, and that inversion is the whole move of the chapter. Once Stead, Storm, and Palm resolve down to tens of nanometers, the question changes. Where it once asked which proteins a synapse holds, it now asks where each one sits and how it moves. AMPA receptors cluster in nanodomains about 80 nanometers across, aligned over the cleft to the presynaptic release sites. A low-affinity receptor is strong only where it stands. Composition was always visible under a lens. Position was the hidden variable, and breaking Abbe's diffraction limit is what turned it into a measurable one. This section explains why the synapse stayed invisible to light for a century, and what super-resolution microscopy let us finally see. The synapse's working parts are all very small. Spine necks are around 100 nanometers, vesicles around 40 nanometers, and the postsynaptic density is under 500 nanometers. Every one of these sits below Abbe's diffraction limit, so for a century light could only blur them. To read their molecular organization, you had to fix and slice the tissue with electron microscopy that killed the very dynamics that carry the function. You could see the structure only by destroying the life in it. Super-resolution microscopy breaks that limit. The three methods are Stead, Storm, and Palm, and the chapter's thesis falls straight out of the break. Synaptic function is the nanoscale positioning and live mobility of proteins, now watchable in living tissue. The wiring diagram was never the whole story. The deeper pattern is this. The instrument's resolution bounds what the object is permitted to mean, cross the bound, and a structure that was mute in life begins to speak. This section explains why a brain is opaque and how chemical clearing makes it transparent. A brain is opaque not because it absorbs light, but because it scatters it. Every interface between cytoplasm, membrane, and protein is a jump in refractive index, and light bends at each jump until the beam is lost. The whole chapter is a variation on one insight from Spalteholz in 1911. Soak the tissue in a medium whose index matches the average tissue index, roughly 1.45 to 1.56, and the internal jumps flatten. Scattering collapses, and the sample turns glass clear. Every protocol in the zoo is one route to that same target. The solvent-based methods are Baby and 3-Disco. The water-based methods are Cubic, Clarity, and Scale. They differ only in how they reach index homogeneity, whether by dissolving the lipids or by swelling the tissue with water. This section explains why brain tissue is opaque, and how a single optical move turns it transparent. Opacity is variance. Brain tissue scatters light because the refractive index jumps at every membrane boundary, N1, N2, N3 packed micrometers apart, and each jump bends a ray. Enough bends and the sample is opaque. Soak it in a medium that matches the tissue's mean index and the jumps flatten. Light travels straight. The brain goes to glass. That single move, refractive index matching, introduced by Spalteholz in 1911, generates the whole chapter. Every protocol, the lipophilic solvents that dissolve the membranes, the hydrophilic cocktails that swell and equalize, is just a different route to the same internal homogeneity. Clearing is what earns volumetric microscopy. It's thick samples. Abolish the index contrast, and photons stop turning. This section explains why. Light sheet microscopy can image a whole brain at cellular scale, where point scanning methods cannot. Point scanning microscopes build a volume, one voxel at a time, generating out-of-focus light everywhere and then discarding most of it. Light sheet microscopy moves the sectioning into the geometry itself. Illuminate the sample from the side with a thin plane of light, detect along a perpendicular axis, and only the fluorophores lying in the detection focal plane are ever excited. A whole plane images at once, so throughput scales with the camera rather than with the number of points. That single move, orthogonal illumination and detection, is why an entire cubic centimeter mouse brain resolves at cellular scale in under a day, where confocal and two-photon scanning never finished even one. This part explains light sheet microscopy, where optical sectioning comes from geometry rather than from rejecting stray light. You illuminate the sample edge-on with a thin sheet of light lying exactly in the detection focal plane, then read the whole lit plane at once through a perpendicular objective. Confocal and two-photon microscopes fight out of focus light by rejecting it point by point, one voxel at a time. Light sheet never creates that light in the first place, since only the in-focus plane is ever excited. Sectioning becomes a property of the illumination geometry rather than a filtering stage applied after the illumination geometry, rather than a filtering stage applied after the fact. Speed follows for free because a full plane images in parallel instead of pixel by pixel. That parallelism is what lets an entire cleared brain be reconstructed in under a day at cellular resolution. This section names what the chapter is for in the book's arc. Optogenetics splits neural control into two independent axes and fuses them in a single protein. Genetics decides which cells carry the light-sensitive actuator, a microbial rhodopsin that turns photons straight into ion flux. Light decides when, with millisecond precision. The surprise is that specificity and timing, which every other method entangles, come apart cleanly here. Pharmacology and designer receptor methods lose because the drug diffuses and cannot be switched off sharply. Light does not diffuse and carries no such lag. One genetically encoded component, addressed by a beam, is the whole idea. Everything downstream is engineering that palette of proteins and delivering the beam. This section explains the structural primitive of optogenetics. Splice one light-sensitive protein into a chosen neuron, and its membrane voltage becomes a function of illumination. That is the whole move. The actuator is a transducer. Photons drive a microbial rhodopsin that either gates open as an ion channel or runs as a pump, converting light directly into depolarizing or hyperpolarizing current. Because light, unlike a drug, does not diffuse, temporal precision is set by the protein's own kinetics on the order of milliseconds. Spatial precision is set by where the gene is expressed and where the beam lands, never by transport. Everything the chapter unfolds is composition on this primitive. Choose the opsin sign, its wavelength, and its speed. Then get enough photons to the target. Control supplants perturbation. This section explains how synthetic optogenetics gives light control to proteins that nature never made light sensitive. Natural optogenetics is stuck with 6 photoreceptors and their evolved chromophores. Every native neuronal channel is blind to light on its own. The move is to factor a chromophore into control, orthogonal parts. The first part is a light-toggled geometric core, almost always azobenzene, which snaps from trans to cis under near ultraviolet light and then reverts under visible light. Light and then reverts under visible light. Conjugate this synthetic switch onto any blind protein, and light now gates it, rapidly and reversibly. That is the whole primitive, a geometry engine bolted to a ligand of your choosing. The surprise is how little coupling this needs. A real chromophore sits inside an intimate contact web, where the subtlest change in its geometry propagates through the entire fold. A synthetic photoswitch has no such demand. It merely has to move nearby. Berlin and Isakov cast this as simple hand movements, a nudge close to the protein that is enough to toggle function. This section explains how optogenetics escapes a scarcity of natural light sensors by taking the photoreceptor apart into pieces you can build on their own. Optogenetics starts from a scarcity. Only 6 evolved photoreceptors catch light, and most neuronal proteins are blind to it. The move of this chapter dissolves that scarcity by factoring the photoreceptor into parts you can build separately. There is a light-sensitive core, an azobenzene or a spiropyrin whose geometry snaps between the trans and the cis shape under 2 different colors. That core is welded to a drug that gates the target and welded to a tether that aims it. Photon catching is no longer a property a protein must have. It becomes a module you bolt on. Once light, sensitivity is a separable bond rather than an evolved chromophore. Any native channel or receptor becomes addressable. The 6 photoreceptor ceiling lifts, and over 40 engineered switches follow from the same decomposition. This section explains how nanoparticles can drive neurons with light without touching the neuron's genome. Think of it as optogenetics without the gene. A transducer placed at the membrane converts a remote light field into the local physical currency a neuron already answers to. The nanoparticle is an antenna. plasmonic gold most of all, that absorbs light and re-emits it as heat, mechanical strain, free charge, or released chemistry, right where an ion channel can feel it. 4 channels, 1 pattern. Light goes in, and a membrane, legible perturbation comes out at sub-millisecond speed. This works because a particle far smaller than its cell heats and cools only its own neighborhood. Bioconjugation aims the antenna at a chosen channel, such as TRP51 or a voltage-gated sodium channel. That buys the molecular specificity the opsin got from genetics. The neuron stays wild type. The specificity moves into the particle's surface. This section explains how a nanoparticle acts as an antenna for the neuron. It captures light the neuron cannot itself feel and re-emits it as a force. The membrane can feel heat, pressure, charge, or a freed molecule. That is the chapter compressed, optical neuromodulation without rewriting the cell's genome, which is the price optogenetics pays. Gold is the exemplar. Its conduction, electrons driven at the localized surface plasmon resonance, shed their energy into the lattice and warm the surrounding water within nanoseconds. The 4 transduction pathways, photothermal, photomechanical, photoelectric, and photochemical, are one primitive scene from 4 sides. A subcellular converter, sized to match the ion channels it perturbs, tuned by particle shape and aspect ratio to fall inside tissue's transparency window of 600 to 1200 nanometers. This section explains how a single question, how freely the animal may move, organizes an entire zoo of optogenetic technologies. Optogenetics promises light-driven, cell-type specific control at millisecond precision. But delivering the right light in the right place at the right time, deep on multiple targets while recording, collapses into mutually exclusive trade-offs the moment the animal must roam. So the chapter refuses to name a best device. Instead, it lays the whole field on a single monotone ladder, head fixed, then fiber tethered, then wire tethered, then wireless. Each rung buys behavioral freedom by paying in something else, where the primitive to carry is this. The field is a Pareto front indexed by degrees of freedom. You do not optimize the interface. You locate your question on the curve. This section explains how a single scalar organizes the entire optogenetics design space. That scalar is degrees of movement, freedom. Optogenetics fixes the biology. A genetically light-sensitive channel gives cell-type specific millisecond control. So the open problem is purely delivery. Shed the right light in the right place at the right time in an unrestrained animal. The design requirements trade against each other, and some are mutually exclusive. They are tunable wavelength, cellular resolution, sub-millisecond timing, deep multi-target reach, simultaneous recording, and free movement. No single device wins all of them. The chapter's move is to linearize that Pareto surface along one axis, how far the light source migrates from the bench toward the animal's head. The rungs run from head, fixed, to fiber tethered, to wire tethered, to wireless, to closed loop. Each rung buys behavioral freedom by paying resolution, power, or noise, immunity. Predict any new device by where it sits on that migration. This section explains the holographic optical neural interface, or HONEY, and the single move that gives it its power. Steer one beam across neurons, one at a time, and you pay in serial time. Shape the wavefront's phase once, and the whole target pattern lights up at once. That is the HONEY move. A phase only. Spatial light modulator holds a computer-generated hologram in the aperture plane. The neural pattern you want is simply its Fourier transform, reconstructed by interference where the light lands. You never draw the pattern. Diffraction draws it, redistributing power into the on spots instead of discarding it at a mask. The trade is exact. Give up direct control of intensity at the target, gain control of phase at the aperture, and buy parallel, scanless, three-dimensional addressing that point. Scanning cannot reach. This section explains how a single computed phase pattern can light up a whole copulation of neurons at once. The chapter's move is to stop scanning a beam across targets one at a time, and instead place light everywhere simultaneously. A phase, modulating SLM, sits in the Fourier plane, so the intensity pattern you want at the sample is just the Fourier transform of the phase you display. Patterning neurons becomes an inverse problem. Solve for the phase mask that generates your targets. Because phase, not amplitude, is modulated, no light is discarded into off pixels. The power redistributes into the on regions. And here is the real prize. It redistributes into arbitrary three-dimensional patterns which amplitude methods cannot reach. This section explains multi-photon nanosurgery, the destructive twin of two-photon imaging. A focused beam stays transparent everywhere except one point. The destructive reaction scales with the square of the intensity, so only the focal voxel crosses threshold. The light entering and leaving deposits nothing. That one nonlinearity is a scalpel with no blade. You sever a single axon, mitochondrion or dendritic spine inside a living brain without scratching the tissue the light crossed. It is the focal confinement primitive of two-photon imaging run in reverse. The deposited energy fragments molecules, an avalanche of quasi-free electrons or reactive oxygen instead of fluorescing. Here's the chapter's place. Nanosurgery is imaging's destructive twin, and controlled injury at single-element resolution is what lets you read the rewiring rules of a neural circuit. This section explains why a two-photon microscope can image tissue and cut it with the same beam. Multi-photon absorption scales as intensity squared, so it occurs only at the focus. That single nonlinearity is the whole chapter. Two-photon imaging exploits it to excite fluorescence in a diffraction-limited spot. Raise the dose 5 to 10-fold, and the same focus becomes a scalpel, ablating one axon, one spine, one mitochondrion, while everything a micron away survives untouched. Nothing about the apparatus changes. Imaging and surgery are one instrument at two exposure settings, the destructive threshold reachable only where the beam converges. So the cut localizes without any mechanical contact. Precision falls out of the quadratic law rather than being engineered in. A needle destroys by touch and cannot confine. The threshold destroys by physics and must. This section explains how diffuse optical tomography images. The working brain. Every functional brain imager reads the same underlying signal, the neurovascular cascade that floods active cortex with oxygenated hemoglobin. The imagers differ only in the physics that transduces that signal. In diffuse optical tomography, the transducer is light. Near-infrared photons in the optical window from about 600 to 1,000 nanometers penetrate centimeters into tissue because the tissue absorbs them weakly while scattering them heavily. Their transport therefore becomes diffusion, and hemoglobin's absorption changes ride that diffuse flux. The chapter's thesis is that a tomographic inverse over many overlapping source detector pairs recovers three-dimensional cortical maps at resolution comparable to fMRI, without magnets and without radiation. The pairs are high density, with first neighbor spacing under 15 millimeters. The contrast is the same. Bold signal that fMRI reads, reconstructed through a photon diffusion forward model instead of magnetic resonance. Resolution is bought with measurement density. This section explains how near-infrared light lets you image brain activity through the skull. Near-infrared light survives centimeters of tissue because water, lipid, and hemoglobin barely absorb across the optical window from 600 to 1000 nanometers. But those same tissues scatter the light hard, so a photon random walks rather than traveling straight. That kills the camera. The reconstruction works in two moves. First, you model the forward diffusion from each surface source and detector pair. Then, you invert the boundary intensities back into interior absorption changes. Absorption tracks blood oxygenation, and blood oxygenation tracks neural firing. This is functional. MRI's bold contrast read optically. This is functional MRI's bold contrast read optically. The chapter's move is this: resolution lost to scattering is bought back by overlap density. When nearest neighbor spacing drops under 15 millimeters, the measurements begin to overlap, and that overlap is the one knob separating high-density diffuse optical tomography from sparse FNIRS. This part explains optical coherence tomography as a way to image the human brain without deforming its three-dimensional geometry. The classical route is histology. Cut the tissue into slices, then image each one. Every slice deforms as it is cut, so reconstructing the volume becomes a losing fight to register those distortions back together. Optical coherence tomography, or OCT, inverts the order. It images the intact block face optically, reading a few hundred microns deep into the surface. Only then does the microtome shave that layer away, and the next face is imaged. Measurement precedes destruction, so the three-dimensional geometry is never deformed to begin with. Because of that ordering, you get histology-grade contrast, the laminar structure of the cortex and the myelinated fibers, across cubic centimeters of tissue and without registration artifacts. The polarization-sensitive variant recovers something more: the orientation of the fibers. That is what lets post-mortem OCT serve as ground truth for diffusion. MRI tractography, the technique that maps fiber pathways in the living brain. The role of this chapter in the book is to place OCT as the distortion-free bridge between two scales. Histology gives micron resolution but destroys the geometry. MRI keeps the intact volume, but at coarse resolution. OCT joins the two. This section explains how optical coherence tomography reads depth out of tissue without cutting it. Back, scattered light carries depth for free if you listen only to the photons that arrive in phase. You interfere the tissue, return against a reference mirror, and only the rays, whose round-trip path matches the reference to within one coherence length, produce fringes. Depth is therefore selected by path, length matching rather than by focusing. Because of that, axial resolution decouples from the lens and follows the source bandwidth alone, on the order of 1 micrometer. That single gate is what the whole chapter is for. It lets you read cortical layers, myelinated fibers, even individual neurons off an intact tissue block before any knife touches it. Think of OCT as histology, with its distortion, sectioning, mounting, staining, and registration all subtracted away, leaving cubic centimeter reconstructions at micron resolution. Everything downstream, whether time domain versus Fourier domain or polarization contrast, is only a different readout of the same coherence gate. This section explains how acousto-optic monitoring reads brain activity through tissue. Light in turbid tissue carries no depth coordinate. Every detected photon is a scrambled path integral, which is why near-infrared spectroscopy and diffuse correlation spectroscopy cannot separate brain from scalp. Ultrasound supplies the missing coordinate. A focused acoustic pulse periodically displaces scatterers only where it passes, stamping its own frequency onto just those photons, tagging them. Because sound travels at a known speed, the delay tau m maps to depth. The depth equals the speed of sound times the delay. Cross. Correlating the detected speckle against the ultrasound code recovers optical intensity indexed by where it came from. The primitive is this. Impose a known carrier on the fraction of a signal you want, then correlate to pull back only that fraction, its delay reading out position. A movable focus becomes a depth gate, and extracerebral contamination falls away. This section explains how a diffuse light measurement of the brain gains the depth it lacks. Diffuse, near, infrared light images brain hemodynamics, but it cannot say where along its path the signal formed. Cerebral blood flow and the pressure passive scalp above it blur into one reading. That blur is fatal when you are monitoring the autoregulation that only the brain performs. The generative move is to stamp a spatial beacon onto the light with a second, sharply focusable modality. A focused ultrasound pulse periodically displaces scatterers only inside its focus, phase, modulating or tagging just the photons that cross there. Cross-correlating the detected speckle against the ultrasound pulse's delay recovers signal from that depth alone balls. Coincidence between an unlocalizable field and a localizable one buys localization that neither has alone. This is the same trick wherever a diffuse measurement is gated by a focused stamp. This section explains how neurophotonic vision restoration works. Blindness from outer retinal disease deletes the transducer while leaving the wire intact. The photoreceptors die, but the ganglion cells and the optic nerve survive, so the move is not to rebuild the sensor. Instead, reinstall light sensitivity directly onto the surviving cells by expressing an opsin in them and then drive them with a patterned projector. The opsin used is channel rhodopsin 2, along with its red-shifted 2, along with red-dis nerve survive. The elegance of this move is compositional. Whatever image processing the dead upstream layers used to perform, an external encoder now computes and pre-applies. So the projected light already carries the code that the target layer expects. Computer-generated holography places single-cell spots of light to deliver it a millier's people, a millier's peep. The prosthesis is therefore sensor substitution plus externalized computation. Replace the one deleted stage in silico, and graft the light gate onto whatever remains alive. This part explains how one form of blindness can be bypassed rather than cured by resensitizing the part of the retina that still works. Blindness from outer retinal degeneration kills only the photoreceptors. The surviving inner retina, its ganglion cells, and their optic nerve projections all stay wired and functional. So the chapter's move is not to rebuild the dead sensor, but to resensitize the intact relay. You genetically express light-sensitive channels in the surviving cells. These are optogenetic probes from the channel rhodopsin 2 lineage. Then you drive them with an excitation pattern that is fed from a camera, computed, and projected onto the retina. The retina becomes its own photodiode array. This is the information bypass. You inject at the first, still living stage of a broken signal chain. Rather, than replacing what died. The structural payoff over electrode implants is this. Light dodges current spread, so it buys stimulation that is implant-less, massively parallel, and potentially at cellular resolution in a regime where electrodes stall near an acuity of 20/550. This part explains why a cochlear implant hits a ceiling, and how swapping its carrier lifts it. The ceiling is crosstalk. Electric current spreads through tissue, so neighboring contacts excite overlapping pools of neurons, and the roughly 500,000 users get far fewer distinguishable pitches than the electrode count promises. The number of contacts flatters you, but the current geometry limits what you actually hear. The chapter's whole move is to swap the carrier. A focused light beam deposits energy without contact and without lateral spread, so each source addresses its own neuron population. The channels become independent, and channel count, rather than current geometry, sets fidelity. That single substitution is the generative pattern. Current becomes photons, and spread-limited delivery becomes beam-confined delivery. Everything downstream is just a different way of delivering confined energy. Infrared heating, optogenetic channels, and optoacoustic pressure waves each supply a different how, and each trades the same axis of selectivity against power. This section explains why a cochlear implant's channel count depends on the carrier, not the electrode count. A cochlear implant ceiling is not how many electrodes you can pack, but how many independent channels the carrier allows. Electric current spreads, so neighboring contacts overlap and the pitch percepts smear. The smearing is worst in noise and in music. The whole chapter turns on one substitution. Swap the diffuse carrier for a focusable one. Light can be aimed, delivered without contact, and confined to a small neuron population, so channel count is set by beam geometry, not by current spread. There are 3 ways photons trip a spiral ganglion neuron. First, infrared heating that depolarizes. Second, light-gated channels, which is optogenetics. Third, light-driven pressure waves, which is optoacoustics. The same primitive runs throughout. Selectivity is bought by confining the carrier, and everything downstream is engineering that confinement. This section explains why brain tumor margins can be read directly from the light the tissue itself gives off. The tumor announces itself in light the tissue already emits, nothing injected, which is what makes the method label-free. Metabolic coenzymes fluoresce on their own. The reduced coenzymes NADH and NADPH, together with the flavins, whose emission ratio enzymes NADPH, together with the flavins, whose emission ratio encodes the redox state, or malignant glial cells rewire their metabolism so that intrinsic signature shifts across the tumor margin. The very boundary that MRI and ultrasound surrender to, once the skull opens and the brain shifts by a millimeter, becomes optically legible in real time, with no contrast agent. That is this chapter's place in the arc. Intraoperative margin assessment, where the standard modalities drift, is resolved by reading autofluorescence. 2 readouts carry it. The first is the steady-state spectrum, which is ratiometric and sensitive to geometry. The second is the fluorescence lifetime, an intrinsic perfluorophore clock that is immune to intensity and to the excitation path, which is what makes it the robust discriminator. This section explains why a tumor can be seen without any injected dye. Cancer rewrites metabolism, and metabolism itself already glows. The brain's own cofactors, NADH and flavins, fluoresce, and their emission tracks the redox state. A tumor perverts. The tissue reports its own pathology with no injected dye. The decisive move is to read fluorescence lifetime, not brightness. Lifetime is a state function indifferent to excitation wavelength, to exposure, to probe geometry. So it survives the surgical field, where absolute intensity, hostage to source, to sample distance, cannot. This buys what MRI and ultrasound cannot: margin assessment in real time, immune to the brain, shift that voids every preoperative image the instant the skull opens. Where the healthy tissue ends, the glow changes state. This section explains why third harmonic imaging finds structure in brain tissue without any stain. A homogeneous medium emits no third harmonic at all. The phase-matching integral cancels to zero no matter how intense the beam, no matter how large the material's third-order susceptibility, Chi3. Signal appears only where the medium changes across the focal volume, and an inhomogeneity about half the size of the focus maximizes it. So the contrast is the structure. THG lights up interfaces, lipid-rich membranes, the roughly 1 micrometer axons and dendrites of brain tissue, because those are exactly where homogeneity breaks. This is why the technique needs no dye. You are not staining anything. You are reading the tissue's own spatial discontinuities. That inverts the histopathology bargain. Instead of fixing, slicing, and staining hours later, the microscope returns an optical biopsy in near real time, contrast for free, wherever the brain stops being uniform. This section explains a label-free optical biopsy, whose contrast comes from a selection rule rather than a dye. Third harmonic generation converts 3 photons into 1 photon at triple the frequency. But in a homogeneous medium, the phase mismatch cancels the process to zero, no matter how intense the beam. Signal appears only where an interface half the size of the focal volume breaks that uniformity. So the picture is a map of the tissue's own discontinuities. Lipid-rich myelinated axons blaze brightest, and structure images itself with no stain, no labels, in near real time. The chapter's role in the arc is to close the gap between an MRI's rough preoperative guess and histopathology's day-later verdict by making the physics of where signal vanishes the diagnostic itself.